Hello, good morning / afternoon to everyone. I am Fernanda Coelho, and I'm happy to share the session with Fabiana Canini, who will talk to you in some minutes. So today is the second day of our session 30, environmental factors driving diversity and composition of fossil and living Antarctic communities. And I would like to thank you all for participating of our session. Today the session has 6 talks, which means that it's going to last 1.5 hours. And the session is being recorded, and the videos will be available in the SCAR YouTube channel. The first day videos are already available. And the discussions on each presentation will occur next week during the conference itself, let's say that. So thanks again for your presence, and I ask Fabiana to present authors of the first talks. Please, Fabi.
Good morning or afternoon to everybody. The first speaker of today is Maria Vernet, and the title of the presentation is The Nitrate-to-the Iron Ratio in West Antarctica Coastal Waters. Please, Maria, when you prefer, you can start.
Thank you, Fabiana. Is this working?
Yes, it is working. It's fine.
Okay. So, hello, everyone. Today, I would like to talk about the nitrate-to-iron ratio in the West Antarctic Peninsula, actually from the peninsula to the Ross Sea. This is a collaboration between several of us at Scripps Institution of Oceanography. And it's an extension of a project funded by the National Science Foundation in the West Antarctica Fjords, the Fjord Eco Project. In 2001, Brundtland et al. Proposed for the California boiling system that the ratio of nitrate to iron in the water column would affect the ability of the phytoplankton to absorb iron. And that would be explained as the iron to carbon ratio in the cell. At high iron concentrations and low nitrate, we would expect a high iron to carbon in the cell, and the opposite is true. And the range that they gave was about 200 for the iron-rich environments and about 10 or lower for the iron-limited environments. So our question was, given that Antarctica is the Southern Ocean is a high nutrient, low chlorophyll environment and is iron limited, what are the nitrate to iron ratios there, and what can it tell us about the phytoplankton and the biogeochemistry? This is a map showing the different data sets that we used. Today, I still don't have all the data, but we have most of them. In the Antarctic Peninsula, there are 3 data sets. In the Amundsen Sea, there are 2 and 2 more for the Ross Sea. So here I'm presenting the nitrate to iron ratio for all the data sets. And there are 3 things I would like to highlight. If we plot the iron to nitrate ratio in the y-axis in a log scale, we can see that the values vary by about a factor of 1,000. And there are 2 families of curves, let's say. One in which the value is less than 10. And these are the 3 near-shore environments in the West Antarctic Peninsula. The red is Margaret Bay from the RATS British program. And these 2 are the fjords in the West Antarctic Peninsula that we studied in Andvord Bay in 2015, 2016. All the rest of the places, the Western Antarctica Peninsula Shelf, the Amundsen Sea and the Ross Sea have much higher ratios, between 50 and 500 on average. I also want to point out that the size of the circles is the amount of phytoplankton. Here is total chlorophyll. So the amount of phytoplankton the two areas can sustain is independent of the ratio of nitrate to iron. If we plot the same ratio in the log scale on the y-axis against iron concentrations, we see that all these ratios fall within a line. That means that the iron concentration in the water column is the driver for this ratio. And there are some interesting features here. One is that although most of the points are along the line, there are some exceptions. And these exceptions are in the Amundsen Sea, in the West Antarctic Peninsula, and in Margaret Bay. And these are stations with lower ratio, meaning they're enriched in iron. And we think that these are locations of iron fertilization, different sources and things like that. The other interesting point is that the 1 nanomolar of iron in the water column seems to divide the two family of curves, the low value, the low ratios, and the high ratios. If we now ask, is this related to phytoplankton composition? I'm going to show 3 graphs of the ratios in the same type of graphs that I showed before for diatoms, for phaeocystis, and for cryptophytes. Phaeocystis and cryptophytes are the two most abundant flagellates in this region. So here the size of the circles is the amount of chlorophyll A associated with the diatoms in this case. And as you can see, diatoms are equally abundant in both regions, and they don't seem to be that much affected by the ratios, although we are still missing, for example, the data for the Margaret Bay. So this is not yet complete. For phaeocystis, we find similar features. The difference is that in the high ratios, phaeocystis is more abundant than in the Western Antarctic Peninsula. Something not surprising, we knew that from many studies. And for cryptophytes, the same thing. It's less abundant overall in the high ratios of the Amundsen Sea and the Ross Sea, and it's higher in the fjords. This is December 2015 in Andvord Bay. So to summarize, we do not find yet a tight relationship between nitrate-to-iron ratios and phytoplankton composition. We think that all the groups have enough plasticity to be able to live in both environments. And here in this graph, we have all the projects for which we have data. This is a log scale of the nitrate-to-iron ratio. And these are the percentage of the different groups, main groups in all the regions. And as you can see, cryptophytes are more abundant in the peninsula, as we knew. The diatoms are mostly everywhere, although richer in this 2011 Palmer LTR January cruise. And the phaeocystis is richer in the Amundsen Sea and the Ross Sea, with less concentration in the Western Antarctic Peninsula. But there is a hint that maybe there is more diatoms here that we knew about, not necessarily related to low nitrate-to-iron concentrations. If now we turn around the question and we ask, do phytoplankton influence the nitrate-to-iron ratios in the water column, we are going to look in detail at some of the data that we collected in the Andvord Bay, in the Fjord Eco project. And here I plotted the iron concentration in the y-axis and the carbon phytoplankton in the x-axis. The green are the stations in which diatoms dominate. Their carbon is more than 40% of the total carbon. And the pink one are the cryptophytes dominating stations. And as you can see, when diatoms dominate, the the concentration of iron decreases much faster as the bloom develops. So if we now plot the same data against nitrate-to-iron ratios in the y-axis now, we can see an interesting feature. Because the diatoms take up iron much faster than nitrate, the ratio increases as the biomass of diatoms increases. That is not true when the cryptophytes bloom. Their concentration is always at the same nitrate-to-iron ratio, meaning that they absorb at the same rate as the environment, while the diatoms strip the water column from iron, and the ratio increases as the bloom of diatom increases. Using this data, we calculated pseudo-uptake rates. So based on the carbon of the phytoplankton and the rate of disappearance of the nutrients in the water column, we calculated uptake rates. So in the y-axis here is nitrate-to-iron uptake, and in the x-axis is concentration of iron. The diatoms are still the green dots, and cryptophytes are the purple ones. And as you can see, the diatoms absorb at a much lower rate than the cryptophytes, meaning the iron to nitrate is higher by a factor about 5. And if now we use the same to estimate iron absorption to carbon, then we see even more interesting data. On the left is the Andvord Bay data, and on the right is the Ross Sea. The Ross Sea has a dissolved iron less than 1, and the WAP Fjord has more than 1 on average. And here is the same numbers, but this is 10 to the 3 and 10 to the 4 dissolved iron. The interesting features are that in the fjord, the iron to carbon uptake goes from 100 to 800. In the Ross Sea, goes from 2 to 3. So this is a difference of 100. And also, the diatoms here in green have a higher iron to carbon uptake than the cryptophytes when in the fjord with high iron, but they have a lower uptake rate in the Ross Sea with low iron. So the plasticity of the diatoms is really something to consider here. If we compare these iron to carbon pseudo uptake rates with the literature, we see that in the fjords, for example, for diatoms is a high rate, as we saw here. But on the West Antarctica continental shelf, the Amundsen Sea and the Ross Sea, these diatom uptake rates are between 2 and 5. So looking at the literature, we see that this correlates with iron-limited diatoms. While the iron-rich diatoms, Marchetti found that they were between 150 and 250 uptake rate. In the fjord is higher, and the California current is also 200. Please keep in mind that the methods are different. So it could be some overestimation here of iron absorption, but it's definitely, even if it's half of it, or it's much higher than in the rest of the other data sets. So now putting both things together, the question is, can the nitrate-to-iron ratios predict the absorption of iron by phytoplankton? And we can see that it does. If we look now at the ratio again on the y-axis and the nitrate and the iron concentration in the x-axis, we see here that in the Amundsen with the blue, in the Ross Sea with the red, and in the WAP shelf with the turquoise, this ratio, and now the size of the circle is the uptake of iron to carbon, is much lower than on the fjords. And we think that the Margaret Bay data set that is here would be similar to the fjords. So definitely the very high iron environments of the nearshore waters that have a low iron to nitrogen ratio have the phytoplankton, a much higher iron to carbon uptake. So to summarize, I have 1 minute. I want to say that the variability of nitrate to iron ratio in Antarctica is a factor of 3. It's driven by iron concentration, and this ratio, the variability of this ratio is about 10 times higher than we see in other iron-rich coastal environments of temperate waters. The phytoplankton, in particular diatoms, can affect the nitrate to iron ratios, and by the plasticity of the diatoms, that they have a low uptake in high nitrate to iron ratios, and they have a very high uptake in low nitrate to iron ratios, they can affect the ratio itself. And in one, they can retain iron in iron poor waters in the water column, and they can retain nitrate in the water column in the iron poor waters, rich waters. This reminds us of the different types of diatoms that exist. And one of them is the type of diatom, the large diatoms that can soak up the iron, slow down, and sink out of the water column. These are the big sedimentation that we see in coastal Antarctica. And one of the examples is the IFEX experiment, in which Ketoceros dicata was seen as sedimenting after iron enrichment. This makes, in a certain way, diatoms to become iron traps. So our initial question of nitrate to iron ratios predicting iron uptake, we can say yes. And this gives us a new tool to now understand iron limitation in the Southern Ocean. Thank you very much.
Thank you, Maria, for your talk. Very interesting. Next speaker is Professor Laura Zucconi, and her presentation is effect of environmental parameters on diversity, community composition, and functional yield and growth form distribution of fungi in Victoria land soils. Please, Laura, do you prefer?
Yes, I'm ready. Do you see it?
Yes, it's ready. Yes, we can see it.
Okay. Okay. Good morning, good afternoon, good evening, good night, everybody you are. Thanks to this CAR 2020 for organizing this meeting online, and thanks to the organizers of this section for the opportunity to show you an analysis on the diversity existing between two different environments in Victoria Land, a coastal one and an inland one, in terms of community composition, richness, abundance, functional yields, and growth forms distribution, and on the effect of environmental parameters on the differences observed. So the area of study is Victoria Land, one of the most investigated areas in Antarctica, thanks to the presence of a wide difference of different terrestrial environments. And the first set of sites is located along the coast and along a latitudinal gradient from the northernmost one, Apostrophe Island, to the southernmost one, Botany Bay. And I can tell you right now that the latitudinal gradient has a very limited effect on the community composition of these coastal sites. A study has already been published, and the differences observed appear to be determined by the different soil physicochemical properties and the type and extension of plant coverage, influencing into the physicochemical soil properties. This is a picture of the collection site at Key Island, and these are some features of the biological soil crust characterizing the other coastal sites with a different type of mosses and lichens coverage. The second set of sites is located in Taylor Valley. Soils from this valley were collected in 5 sites from 3 different localities close to the 3 lakes present along the valley, Frixel, Hoare, and Joyce lakes. And the soil samples were collected close to the lakes, but far enough to avoid the water environment, the influence of the water environment and the influence of the microbial mats characterizing the ages of the lakes. This is a picture of the landscape of the area, and these are some pictures of the soils that we have collected and analyzed. So the soils from the coastal sides and these other soils, inner soils, were both studied through IPS1 method coding sequencing and compared and analyzed in the light of the different physical-chemical soil properties. Our aims, so, were to characterize the fungal communities associated with these 2 different environments, the inner one and the coastal one, and to relate the diversity, community composition, functional yields, and growth forms distribution to the soil physical-chemical properties. We obtained 860 OTUs, and the taxonomical composition of this site agrees with data already reported from other authors for other substrates and other localities. So we obtained the prevalence of Ascomycota and Basidiomycota, followed by Chytridiomycota and Mortierellomycota. A further interesting datum that is not new is the absence of a core community. Neither for the coastal sites, nor for the inner sites, and nor for all the sites as a whole. So no species was found in almost all the samples collected. Anyway, some taxonomic differences are present between these 2 different environments. For example, the richness of Ascomycota is higher in coastal sites, and these higher values agree with those recorded for the 2 main classes recorded within the Ascomycota, Eurosomycetes and Ophidiomycetes, already reported as the 2 main classes within this phylum by other authors, while differences were observed in for Chytridiomycota with higher richness values for the inner side. We grouped the OTUs into different growth form categories, namely filamentous fungi, black yeasts, and yeasts, and these latter were also observed in terms of Ascomycetes and Basidiomycetes yeasts separately. We did this as a meaningful tool to highlight adaptive strategies to the Antarctic constraints. In fact, filamentous fungi, filamentous growth allowed to explore a greater surface to search water and nutrients, and makes possible to form aggregates able to retain water and nutrients, especially there where the soils are unconsolidated, as in inner sites. Soil inhabiting yeasts are known to be well adapted to polar environments, thanks to many peculiar morphological and physiological characteristics. And under permanent stressing conditions, black fungi, also known as mycocolonial or meristematic black fungi, show a set of adaptation to a wide variety of stressing conditions. Differences are present for richness of black yeasts and of yeasts. And in this last case, the differences observed are mainly due to the differences within the Ascomycetes yeast group. And bacterium acids are already reported as prevalent in polar environments, but in the recent year, a greater number of different ascomycetes has been recorded using specific culture media, and their greater richness in inner sites is probably due to their greater serotolerance, and this is in agreement with the lower content of water characterizing the soils of the inner sites. When we go to see for the values of the evenness, the first interesting result is a low evenness for the total community. So a low evenness value suggests a low ability to react to changes of the environmental conditions, so a low resilience ability. A second interesting result is the presence of differences in almost all the cases here reported for all fungi and all the other growth forms, with higher values for the inner sites, with the only exception of ascomycetes, suggesting a higher resilience ability for the inner sites compared to the coastal ones. Interesting is the high value of black yeasts in inner sites, that is close for evenness to 1, suggesting a high resilience ability. Black yeasts that in the previous slide, as you can see, appear to be characterized by a low richness value. So for this growth form, possibly a lower number of species is associated to species that are more uniformly distributed, and this confers to this growth form and higher resilience ability. When we go to analyze the functional gills distribution, 2 are the main functional gills that have been observed. For these are lichenous fungi and saprotrophs, and differences were not observed for richness and abundance of these 2 main functional gills. But when we go to see for the evenness, we obtain again, as before, higher values for inner sites compared to the coastal sites. We correlated abundance and richness of the main phyla of the growth forms and of the functional gills with the physicochemical soil parameters. And all parameters have a greater effect on richness than abundance. And this is the reason why I'm showing you only the data concerning the richness of these groups. And among these, the most relevant are pH, potassium, and carbon content, with different effects, positively or negatively, among the categories analyzed. The composition of the total fungal communities and the different growth forms categories are well differentiated among the localities, and this result agrees with what I told you at the beginning. So it's in agreement with the absence of a core community among the localities studied. We have considered the effect of the different parameters on the variance of the community, and the pH, carbon, and potassium content have a prevalent effect in determining the composition of the Taylor Valley and inner and coastal side samples, but among these, pH and potassium are more determinant in conditioning the composition of the inner sides, while the carbon content is more important in determining the composition of the coastal sides, these ones that are reported in green. In addition to these 3 main parameters, even soil granulometry is influenced. You can see here in bold are reported those data that are more relevant. And among these, the almost always higher percentage of sand in all samples result in a lower ability to retain nutrients, carbon, cations, and water. The differences above reported are also evident when we analyze the functional yields, and even in this case, potassium and pH are more determinant in conditioning the community composition of the inner sites in blue, while carbon is prevalent in determining the composition of the coastal communities. So to conclude, which are the main results? First of all, a low evenness for the total community, suggesting a low resilience ability to external changes. Then a higher evenness for almost all the groups in inner sites than coastal sites, suggesting a higher resilience ability of this latter of these former sites compared to the coastal one. Then communities well differentiated between the two environments in agreement with the absence of the core community. The absence of the core community neither for coastal nor for inland sites nor for all the sites as a whole. A taxonomic composition confirming results reported from previous authors with the prevalence of Ascomycota and Basidiomycota, and within Ascomycota of the two classes of Dotidiomycetes and Eurasiomycetes, and finally the presence of a lot of undetermined sequences, suggesting the importance to continue to study this environment even with cultural approaches in the light of the bioprospecting. Thank you for your attention, and thanks to the Italian Antarctic Research Program for funding this research. Bye.
Thank you, Laura. And then next speaker is Sabrina Heiser, and the presentation is Connectivity of the Tidal Red Seaweed, Procamium cartilagineum, and how it affects the distribution of defensive secondary metabolites.
All right, thank you very much. Can you see the presentation?
Yes, we can see it.
Okay, thank you. So thank you very much to the organizers for inviting me to give this talk. I'm really excited to be part of this online meeting and to be able to present part of my PhD research. So the overarching question that I would like to discuss today is, how are ecosystems structured? And we can address this question by looking at the general species diversity within ecosystems and investigate how these species are distributed and basically answering who is where. But it is not just species diversity that we need to worry about. Something that we understand even less about is intraspecific diversity and the distribution of those organisms. So answering the same basic question as before, as of who is found where, but at a slightly different level within the ecosystem. And intraspecific diversity is just as important as species diversity, especially when it comes to the resilience of populations and ecosystems towards changes within the environment. And I'm studying those kind of questions in the context of macroalgal forests around Parma Station, which is up here on the Western Antarctic Peninsula. And at Parma Station, we still get these really dense and lush macroalgal forests because we still have enough sunlight during the year. And the biomass of those macroalgal forests is comparable to that of giant kelp forests that many of you can probably find on your own coastline, which means that those macroalgal forests are incredibly important to the ecosystems as primary producers, but also as ecosystem engineers. So they are providing habitat for a lot of other organisms to live in. And at Palmer Station, we have these big brown canopy-forming seaweeds that are sort of dominating those macroalgal forests. And then underneath and between those, we have a dense understory cover of red seaweeds, which are either bladed or finely branched. And my study organism, Plocamium cartilagineum, is one of those red seaweeds. So worldwide, we know that in general, there's a lot of cryptic diversity within Plocamium, which is why cartilagineum is in quotes, because that's the name we have for it in Antarctica at the moment. But we do indeed know that it's different from the Plocamium cartilagineum that has been described elsewhere. We also know that there's a very high secondary metabolite chemodiversity within populations, meaning that different individuals within the same populations are producing different chemical defenses against their grazers. And the question is, what is the evolutionary importance of this chemical diversity? So how is it distributed between populations and what's maintaining it? And what we know about it already from before I started working on this project at Palmer Station makes it an ideal system to study those kind of questions. So back in 2012, Plocamium was sampled from around Palmer Station, and they collected 21 individuals with 3 replicates from 3 different sites, from 7 different sites. And they described those individuals for their secondary metabolite diversity. So in order to get at what the chemodiversity was like within around the area. And then they also sequenced the RBCL and the COX-1 genes in order to determine whether there's any cryptic species diversity going on. And what they found was a total of 5 different chemogroups. So a chemogroup is basically a unique mixture or a cocktail of secondary metabolites, because an individual doesn't just produce one defensive chemical, but a mixture of different chemicals as a defense. And throughout the rest of the talk, I'm going to color code the different chemogroups in order to visualize them. And they also found 2 different haplotypes by doing the single gene sequencing, which means that individuals were genetically different from each other, but not different enough to be different species. The other thing that they observed was a very high site specificity for the chemogroup distribution. So certain chemogroups were found at certain sites. And the other thing that they identified was that certain chemogroups were produced by individuals within certain haplotypes. So we seem to have a very strong geographic distribution of those chemogroups, and we also seem to have some underlying genetic difference between the individuals. And this is where I kind of joined the project and started working on it. And one of the questions that we are interested in is whether the patterns of genetic differentiation can be correlated with the chemogroup differences that we are seeing. So for this project, we had 3 field seasons, 3 summers between 2016 and 2018, during which we did a lot of sampling. And one of the things that we identified during the sampling was that we found a total of 19 different chemogroups. And I just want to point out here that a PhD student at the time, Andrew Schilling, is the one who did all of the chemical analysis on the project. And then I did the single gene sequencing for the COX-1, and for some of the individuals, I also did the RBCL in order to identify the haplotypes that those individuals belong to. And this is the network of the haplotypes. And as you can see, I found the original haplotypes, haplotype A and haplotype B, that had previously been identified, and they were certainly the most abundant ones as well. And just to orient you, I used 2 different main colors. So the different shades represent the 19 different chemogroups, and I color-coded all the individuals almost for the individuals that fall into haplotype A with the purple color, and the individuals with different chemogroups that belong to haplotype B with the green color. And as you can see, haplotype B is a lot more chemically diverse than haplotype A. And I also identified a couple of more haplotypes, C through F, which are only one base pair different from haplotype B, and they were producing the same chemogroups that were also found in haplotype B. But overall, in order to get at the question that I posed earlier, whether patterns of genetic differentiation can be correlated with the chemogroup differences, we needed a lot more fine-scale data to look at the genetic differentiation of those individuals. So for that, during our last two field seasons in 2017 and 2018, we conducted transects perpendicular to the shore in order to have a far more fine-scale geographic sampling. And where possible, we conducted 3 transects per site that were 30 meters apart from each other. And then we collected 5 individuals from each of the 3-meter depth intervals. And I just want to point out here that that was our goal, but obviously we couldn't always find enough individuals at each of the depths, so sometimes we had a few less individuals. These are the 5 sites that we were able to run 3 full transects at around Palmer Station. So we have got a couple more sites where we were able to do partial transects, but I'm not going to show you the data for those today. Down here, we've got the different chemogroups that we were able to find at those sites. And as you may notice, some of the chemogroups are missing here, and that is simply because we didn't find those particular chemogroups at those sites. And then we also have chemogroup 4' down here, which we called 4' because originally in 2016, we described the first 12 chemogroups. And then as the field seasons went on, we found more. And chemogroup 4' simply clustered closely together with chemogroup 4. And then we have, I indicated the haplotypes down here. So haplotype B is the one that's a lot more chemically diverse. And then haplotype A only has chemogroup 12 in it for those individuals here. And this is our first site, East Litchfield. So just to orient you how I'm presenting the data here, I collated all of the 3 transects together on one graph. And then on the y-axis, we've got the depth distribution of the individuals. And on the x-axis, we've got the number of individuals for each site. And as I mentioned earlier, we weren't necessarily always able to find the 5 individuals for each of the transects. The other thing that I wanted to point out here as an example is that we have somewhat of a depth distribution going on of the chemogroups. So the shallower depth appeared to be dominated by chemogroup 12, then chemogroup 4' is becoming more dominant, and in the deeper waters we have primarily chemogroup 1 present. And we had a similar sort of depth pattern also at Nozzle Point, and also at Laggard over here. And then we also had a couple of sites that had were very specific in the chemogroups that they were producing. So Southeast Bonaparte over here, for example, was mostly dominated by chemogroup 4. And in order to get at the question of whether the genetic differentiation can be correlated with these the distribution in the chemogroups that we're seeing, I am currently still in the process of developing microsatellite markers. And what I can do now is show you some of the preliminary data for those markers that I've already developed and for which I analyzed some samples from Nozzle Point and Stepping Stones, which were 2 of the sites that were nearly the furthest apart from each other. So this is the principal coordinate analysis of the multi-locus genotypes that were determined using the 6 microsatellite markers that I've developed so far. And just to orient you here, the further apart the points are from each other, the less genetically similar they are to each other. And then down here, we have the different colors indicating the different chemogroups again, and the different shapes are our 2 different sites that the individuals came from. So first of all, we've got haplotype A or chemogroup 12 that is clustering down here. So that was quite nice to see that the individuals that we had identified to be in different haplotypes separated or clustered independently from each other, showing that that sort of genetic differentiation is continued with our multilocus genotypes. But the thing that I got really excited about was when I started looking at the data and saw that the individuals up here that were producing the same chemogroups but are from different sites are more genetically similar to each other than the individuals from the same sites with that are producing the very different chemogroups. So chemogroup 12, the haplotype A, is clustering very separately from those. So I'm excited and I'm looking forward to finish developing my microsatellite markers and looking or seeing how these patterns are going to continue as I analyze more samples from more sites as well. So coming back to the original question, whether patterns of genetic differentiation can be correlated with the chemogroup differences. So far, we've seen that individuals separate into the different haplotypes when we use the multilocus genotypes, and that those from different sites with the same chemogroups clustered together. And then overall, we are hoping that with these results, we can further unravel what is driving the distribution of the chemogroup diversity within and between sites. And that is important because Plocamium is a very abundant and widespread member of Antarctica's macroalga seaweed or macroalga forests. So it is very important to the local ecosystem, and it will also more generally inform us about the connectivity between seaweed populations that are crucial for the ecosystem. So with that, I'm looking forward to any potential questions or discussions next week, and would like to acknowledge our support that we had on station, the field teams, of course, my PhD committee members, and all of the funding sources. So thank you very much.
Thank you, Sabrina. And next speaker is Julie Schramm, fatty acid transfer from Antarctic algae to the benthic amphipod Gondogenea antarctica. Please, when you're ready. You have to turn on your microphone.
Yes. Thank you. Okay. Can let's see. So can you see my first slide?
Yes, we can see it.
All right, great. Thank you. I am really excited to get to share some of the results of our recent work in Antarctica on the fatty acid trophic transfers to the primary consumer Gondogenea antarctica, which throughout this talk, just for short, I'm going to be referring to as Gondo. So I am really interested in the interactions between primary producers and primary consumers. And here in this picture, this is an example of some of the consumers and producers I really like looking at. So on this red seaweed, you can see there are a bunch of little amphipods hanging out, but it's hard to tell how they're interacting here, whether these amphipods are consuming this seaweed or they are just hanging out for a little while. So often in these kinds of cases, you need to go to a microscope to really get an idea of what is going on with these small primary consumers. And as you can imagine, this can be difficult to get an idea of what's happening at a community or population level. And so the method that I have been using recently to better understand the dynamics between primary producers and consumers is to use biomarkers. And more specifically, I've been using fatty acids as trophic biomarkers. So fatty acids are the building blocks of lipids, and a lot of, or the essential fatty acids, so the fatty acids that animals cannot produce on their own, are produced by primary producers like the seaweed or diatom in this diagram here. And so these essential fatty acids are consumed along with the rest of the tissues of an organism, and those fatty acids get incorporated into the tissues of the new organ of the consumer, and those get passed along up the food chain. However, we are not exactly what we eat. We can look similar and we can incorporate some of these building blocks, but we never have exactly the same profile. And when I'm talking about the profile, I mean that those primary producers have some profile or fatty acid composition that is then consumed by a consumer, and through the process of metabolism, reproductive investment, or somatic growth, there's some kind of conversion or synthesis where these fatty acids are changed, but then over time, some of those fatty acids are retained. And so we can use the retention of those fatty acids to help us figure out who is eating what. And so where I have been performing this work, or looking at, the area that I'm interested in is along the western Antarctic peninsula, where in the same region that Sabrina was just discussing, where between 5 and 40 meters along the nearshore benthos, the bottom is covered by large stands of macroalgae, including these big canopy-forming brown macroalgae. And there is often an understory of these smaller red macroalgae. And within these big canopy-forming macroalgae, there are often lots of primary consumers. So I'm showing you this image here to highlight one of those species, Desmarestia. And within this picture, there are a lot of primary consumers, mainly amphipods. So if you haven't already picked out some of these amphipods that are hanging out here, I've highlighted them. So these amphipods really like hanging out on the Desmarestia because it does produce a lot of secondary metabolites that make themselves taste bad to many organisms. But the amphipods are not actually consuming these big macrophytes. So the macrophytes or seaweeds are producing chemical defenses. They protect the amphipod, or they protect themselves against predation or consumption by the amphipods. But the amphipods hang out there because the seaweeds do produce those chemical defenses, and the amphipods sort of eavesdrop on this chemical defense and obtain a refuge from predation. And while they are there, they consume the epiphytic diatoms or filaments that are on the macroalgae. So they remove some shading. So epiphytes are anything that live on the seaweeds. And this relationship has been termed a community-wide mutualism. So we know that this is happening, that this relationship exists. However, we also know that these amphipods don't just eat the diatoms. We know that as these big brown seaweeds die and then start to degrade, so do their chemical defenses. And when they're degrading, these amphipods will consume these tissues. They're no longer protected by these chemical defenses. We also know that the amphipods eat the epiphytic diatoms, and they will consume some of these understory red seaweeds. But we don't know how often this happens or how prevalent these different food sources might contribute to an amphipod's diet in the wild. And that is what we wanted to investigate in the study that I am presenting today. So we had two basic questions. The first one is the amphipod Gondogenea antarctica what it really eats after 9 weeks in the lab? To test this, we performed feeding assays where we fed amphipods monospecific algal diets. So we gave them one food that they could consume over the 9 weeks in the hopes that they would pick up the fatty acid signatures of their diet. So we would know exactly what that trophic modification would look like when they're consuming one diet. And then we compared the similarity of these the fatty acid profiles of the amphipods on the diets and their diets using multivariate statistics. Our second question was whether or not Gondogenea antarctica consume any of our experimental diets in the wild. And so to figure this out, we compared the fatty acids of Gondo collected in the wild and compared those to the Gondo that we maintained on our monospecific diets. So all of this work was performed on the Western Antarctic Peninsula on Andvord Island at Palmer Station, where we would go out in the zodiacs and collect all of our samples by hand on scuba. So each of the seaweeds that we collected, so here you can see some of those big brown macrophytes. So this one is Hemanthothallus grandifolius. We collected all of these by hand in the field, and we also collected our amphipods from the field. And the way we did this was by targeting the algae that they live on. So we would collect these Desmarestia species in these fine mesh bags so that we would capture any amphipods that were residing on those seaweeds, and then brought everything back to the lab where we thoroughly rinsed all of the amphipods off of the seaweeds that we collected and picked out our target species, Gondogenea antarctica. And then we collected tissue samples from all of the seaweeds that we were going to use for our feeding assays. We collected live or initial samples from each of the species. And then for those species that are strongly chemically defended, the Desmarestia anceps and Hemanthothallus grandifolius, we freeze killed those algae and so that we could feed them to Gondogenea, because otherwise they won't eat it. We then maintained groups of Gondo on these diets for 9 weeks, after which time period we collected samples for fatty acid analysis. All the tissues for fatty acid analysis were extracted for their lipids and fatty acid methyl esters at the University of Oregon. We then ran the fatty acid methyl esters through a gas chromatograph mass spectrometer, which kicked out these chromatograms from which I was able to identify all of the constituent fatty acids from each tissue sample, which I was then able to use to build a fatty acid profile. So the fatty acid profiles I'm going to be discussing are all of the fatty acids I was able to identify in each of these samples. So here on this NMDS plot, I have the data. Each of these symbols represents one replicate, and that replicate is made up of, it's a multivariate point, so it's made up of all of the fatty acid methyl esters or fatty acids that I was able to identify in each sample. So points that are closer together are more similar to each other than those points that are further apart. You can see that some of these symbols are filled in and some are empty. Those represent those initial or freeze-killed samples, and then they're color-coded by species. The triangles represent the algal diets that we fed the amphipods, and the circles represent the amphipods. And then on top of all of this, I have plotted vectors that indicate the fatty acids that are contributing to the differences or the distribution of these samples. And I'd like to highlight some fatty acids that are commonly used as diatom red algae and brown algae biomarkers. Not everyone always is able to test that these biomarkers truly are biomarkers for these things, but in this case, we are able to do that. And you can see that the fatty acid biomarkers that are commonly used for each of these species are driving some of the distribution. So here we've got brown algae, and these fatty acids here are often used as biomarkers for brown algae, the same with the diatoms and the red algae. I would like to also point out here that the different species are grouping together. So we have the diatoms, the Hemanthothallus and Desmarestia and Palmaria all grouping together by species. However, like I pointed out before, even though you eat something, you don't necessarily look like your diet. So our amphipods are falling kind of in the middle of this resource polygon, which is what this shape here is. So they are within this space, so it's showing that amphipods did consume something within these potential diets. So our amphipods grouped very closely together, and they're inside our resource polygon, which we would expect with the amphipods on our monospecific diets. These black circles are the wild amphipods, and it was very gratifying to see them fall inside our resource polygon as well, because that means that we are identifying likely diets for these amphipods. It also appears that a large constituent for their diets are these diatoms, because they're grouping the most closely to those amphipods that were fed diatoms. So to get back to our initial questions, is the amphipod Gondogenea what it eats after 9 weeks in the lab? And we found that yes, the Gondogenea picked up the fatty acid profile markers from their diets, and they did differ from the wild amphipod population. So we know that they are not just looking like what they eat in the field, they are representing our diets that we fed them. And that the common biomarkers that are often used to highlight or identify diatoms, red algae, and brown algae are coming through in our consumers. And then our second question, is Gondogenea consuming one of our likely diets in the wild? Because the wild amphipods were falling within that resource polygon, it does appear that we are identifying likely diets of wild amphipods. So you might be asking yourself, what do we do with this information and where do we go from here? We are continuing on performing more feeding assays in which we are using more macroalgal species. We want to see how these additional macroalgal species are trophically modified by amphipod consumers. And we are expanding the number of amphipods that we are analyzing so that we can get a better idea of how amphipod communities might be trophically modifying their diets. And we're going to use these resource libraries that are generated by these feeding assays to better understand the trophic connections of wild amphipods along the Western Antarctic Peninsula. We recently did a cruise along the Western Antarctic Peninsula where we collected macroalgal associated invertebrates and macroalgae to get an idea of how they change along a sea ice gradient and how those connections might be influenced along that gradient. So this information will be directly used for that, better understanding those connections. And so with that, I would like to acknowledge and thank all of the support we receive on ICE and the funding sources. Thank you.
Thank you, Judy, for your presentation. And the next one will be Roland Beetle, the evolution of benthic invertebrate community ecology in the Cenozoic of Antarctica. Roland, please, when you're ready.
Okay, can we all see that?
Yes, we can see it.
Okay, thank you very much for having me talk in this session. I'm today talking about benthic vertebrate ecology through the Cenozoics, that's 66 million years ago to the present day. So starting in modern Antarctica, the Antarctic benthic community structure is unusual in modern Antarctica compared to the rest of the world because it is dominated by epifaunal suspension feeding organisms. And a lot of these are stationary groups, such as brachiopods, stalk crinoids, and sponges. In addition, the Antarctic community structure lacks shell-crushing predators. So things such as lobsters and crabs are missing from the community, as well as things like sharks and rays. Now, how did this ecology evolve? And this is the question I'm trying to answer by looking at the fossil record of the Cenozoic. So we're going to start by looking in the Eocene. And this is work published by Aronson et al. And it's some of the only work that's been done on ecological structure in the fossil record in Antarctica. So the theory that he proposed was that in the Eocene, we have this cooling here towards the Middle Eocene. And at this time, the number of predatory crabs reduced because of the cooling. Crabs don't like cold water. And because of this decrease in predation, we have preserved on one place in Antarctica dense assemblages of stalked crinoids and brachiopods at this time period. So this has been suggested to be the time period when the modern Antarctic ecology evolved. So as I said, I've been looking through the Cenozoic, and the first thing that happened in the Cenozoic is 66 million years ago, between the boundary between the Cretaceous and the Paleogene, there was a mass extinction, the KPG mass extinction. This is one of Earth's 5 big mass extinctions in Earth's history, and it's the last one before modern animals arose. It occurred, as I said, 66 million years ago, with a worldwide loss of 40% of genera and 76% of species. Now, in Antarctica, we have looked at the extinction metrics and realized that the metrics for genera and species extinction based on mollusks was as high as the rest of the world. And this was work done by James Witts in 2016. So the Antarctic fossil record shows that there was a massive extinction even in Antarctica. So work we have been doing on this mass extinction horizon has been on Seymour Island off the northeast tip of the Antarctic Peninsula. And the extinction horizon in Antarctica runs north to south up the island. It's a very obvious horizon, and it's very well exposed. This shows the extinction horizon in the field, and it forms a very prominent scar slope across the island, looking north up across the island. And on the left-hand side, we have the Maastrichtian, the end of the Cretaceous, and on the right-hand side, the Paleocene, the start of the Cenozoic. And as you can see, these are really well-exposed sediments, are all lightly dipping. So it means that we can walk through time very easily, collecting fossil samples at set intervals to look at biotic change over this extinction horizon. What did Antarctica look like at this time? Well, this is the James Ross Basin where the Seymour Island sediments were deposited. It's a shallow water basin, outer shelf shallowing up to deltaic into the Paleogene. And in the background is the volcanic arc of the Antarctic Peninsula, which was covered in active volcanoes, covered in forests. And we had big animals that we found in the sediments here, including this large crocodile-mouthed mosasaur, and sharks, and fish, and long-necked plesiosaurs. And some of the weirder invertebrates that we get are ammonites. So ammonites are a type of cephalopod that became extinct at the KPG boundary. And in this section, we get these weird uncoiled ammonites, they're called heteromorphic ammonites. And they can be up to several meters long, so really weird things. But we also get normal coiled ammonites and nautiluses, as well as mollusks and crinoids. So what we've been doing in this section is looking at how ecology changed over the mass extinction horizon. And in this diagram, each line represents a species of either gastropod or bivalve, and the color represents a mode of life. So this is a combination of feeding, tearing, and motility. This is the sedimentary log, and where it just shows you what rock types they were. The KPG boundary is represented by the dotted line, and the black dotted line represents a change in environment from outer shelf to deltaic. So immediately you can see there was an overturn in species at the KPG boundary. And it seems to be more pronounced in epifaunal groups, especially slow motor epifaunal carnivores. Things that are infaunal actually seems to have survived fine immediately after extinction, then carried on through. Whereas this epifauna seems to have overturned, particularly in the gastropods. So there is a period where we have a slightly different ecology, but then things seem to come back, different species, but with the same ecological groups, ecological niches. So looking at tearing, motility, and feeding in more detail for the different parts of the section. So in this diagram, this is the Cretaceous, this is the KPG boundary, this is the first Paleogene, and this is the second Paleogene when the environment changes. And the first set of diagrams is based on species numbers, and the second on absolute abundance of the number of specimens. And they're all standardized for relative abundance. So looking at the species numbers, we can actually see that there isn't much change over the KPG boundary and into the further up in the section, interior feeding or motility. The change is shown in the absolute abundance of specimens because immediately after the KPG boundary, the unit is dominated by 1 or 2 species of really high abundance taxa, with 1 or 2 single specimens of epifauna. But then this starts to normalize back further up the section and recovers further up through the section. So we can conclude that there are changes in ecology over the KPG boundary, but using these MDS plots, we can see that the mass extinction did not cause a major shift in ecology. So these MDS plots are based on absolute abundance, A, and species numbers, B, and each point is a sampling horizon, but it's the ecological composition of that sampling horizon, the proportion of each types of ecology. And there's quite a lot of overlaps in the diagram, showing that there wasn't that much difference between the Cretaceous and the Paleogene ecologies. So having shown that this first unit in the Antarctic fossil record doesn't really show any changes in ecology with a mass extinction event, we went on to look at some of the key groups that have been used to try and understand predator and prey relationships in the fossil record. So we decided to look at stalk crinoids because these were a very key organism used to establish when the modern Antarctic community structure might have evolved. And also because they're very affected by changes in predation. So stalk crinoids generally today live in deeper water. They avoid shallower waters where there's higher crab predation. So if you're finding crinoids in shallow waters, it suggests that they're safe to live there. There isn't any crabs. And as I said, their presence in the Eocene has been linked to cooling climate and lowering crab predation. So we documented the fossil record of stalk crinoids for the whole of the Southern Hemisphere. This included new samples, including 3 new species from Antarctica and from sites all over southern Australia. And this just shows some of the new species that we're describing at the moment. We also documented all the literature occurrences we could find. And this, as I said, was for the whole of the Southern Hemisphere. The numbers on this diagram show you the localities where we have fossils from, and these correspond to numbers in the diagram, which are plotted against the time in which they occurred, and they all occurred in shallow water. So the main point to take for the Antarctic fossil record here, in number 1, is that they existed before the Cenozoic, and then we found new specimens in the Paleocene and the late Paleocene, and then carried on to the Lamaceta Formation. Oops. And these are the specimens that form the theory of the evolution of the ecology in the Eocene. But what we're showing is that the Antarctic fossil record actually had shallow water crinoids all the way through until that time. So there's something else going on here. And actually in the Southern Hemisphere, shallow water crinoids are found all the way to the early Miocene in New Zealand. And the ones in Australia follow a pattern of migrating through the continent as the basin opens between Antarctica and Australia at this time. So some other work we've done has been to look at the key predator thought to have affected these ecological structures. And this is decapods in so crabs and lobsters. So this diagram, the Cenozoic period we're interested in is shown in red, and they're all different along the bottom families of crabs and lobsters. On the right-hand side, this just shows you where there are fossil records in Antarctica, and the red ones show which ones have crabs in them. And these show you the extent of the fossil record of the crabs. So what this diagram shows is that we have lots of decapods in the Eocene, and they go all the way through the Eocene. We have decapods in the early Miocene, and we even have occurrence right up in the Pliocene much more recently. But what is important is that the decapods actually are quite diverse throughout the entire Eocene, so we do not see evidence for decreasing predation or decreasing crab numbers. So just to look at these key predator and prey relations, so as we said, the stalk crinoids are found all over the Southern Hemisphere from the Cretaceous to the Miocene in shallow water, not just in the late Eocene when it starts to cool. And that there are decapods in the fossil record in the Eocene, the early Miocene and the Pliocene, so almost quite recent fossil records. And a point that's interesting about these groups is neither of them are common as fossils. So the fact that we're finding them means that they were probably around much higher abundance. They both have multi-element bodies. They have lots of different parts. And when they decompose, they actually disarticulate and fall apart after only about a day or two in the fossil record. All the soft tissues dissolve and they just fall apart. So they don't normally get preserved that easily. So the fact that we're finding them means that that they're in more abundance. And in some of the localities, including the Seymour Island localities, the numbers of them suggest that there might be preservation, might be a factor in their occurrences. It might be some sort of exceptional preservation going on at this site. But overall, we can say that this Eocene cooling did not seem to restructure the ecology. So the last section of the talk is about some work that I've just started doing recently on King George Island. So the localities we're looking at here are date from 29 to 22 million years ago. And we're asking the question, did the glaciation change the community structure? So obviously the formation of the Antarctic ice sheets, these deposits are the first deposits that have evidence of animals living under icy conditions. And this evidence includes diamectites, striated rocks and drop stones. So they were deposited, the sediments were deposited in a glacial environment. So we did some work on the South Shetland Islands at two localities, Lyons Rump and Cape Melville. And first of all, we're going to talk about Polynez Cove formation about 29 to 24 million years ago. And this is my field assistant, Anna, on the edge of the cliff. We're sampling this horizon here, which is absolutely stuffed with fossils. And that's Lyons Rump in the background. The weather wasn't always this good. In fact, I think it was one of the only sunny days we had. And preliminary results from the field investigations show us that the units are dominated by scallops, such as these ones, or brachiopods, which were described by Bittner et al. In 2009. Both of these groups are epifaunal suspension feeding groups. So it is similar to modern Antarctic community structure. And that seems to be like a nice conclusion, that as soon as the glaciation happened, you get this modern Antarctic community structure. And there was very little evidence for any predators found. We didn't find any evidence of crabs in this unit, and we didn't find many gastropods at all, but not many evidence for predators at all in the sections. Unfortunately, it's not that simple, because there's a problem to this. In the next unit we looked at, the Cape Melville Formation, which is younger and also deposited under ice conditions 22.6 billion years ago, is providing a different story. Now, all the units so far have been deposited in more or less shallow waters, and the Cape Melville Formation differs in that it was deposited in slightly deeper waters. And this is a picture of the Cape Melville Peninsula looking back towards King George Island. And as you can see, it's a really steep cliff, which is a real fun adventure trying to get up there to do our sampling on a very snowy day. So this unit, unfortunately, completely complicates the story that we've been looking at. And this is because it is dominated by infaunal deposit-feeding bivalves, such as nucleonids and nucleolids. We do have some predatory gastropods like metabolite, but importantly, we also have very large numbers of predatory crabs in this unit in an icy environment. So this is as I said, making the story much more complicated. We've looked at the basic composition of the community. We've taken out all the soft-tissued animals that wouldn't preserve, and then compared the proportions of the groups to modern communities in South America and Antarctica, just to see what's going on. And the Antarctic one is here, the fossil one, the Cape Melville Formation, and then we have 3 South American localities here, and then Antarctica here. So as you can see, this fossil community is actually more similar to South America than it is to the modern Antarctic community. So overall, it looks like glacial marine conditions may have caused the onset of the modern Antarctic community structure, but looking at the reconstruction of the Cape Melville Formation with the predatory crabs and even lobster and lots of informal bivalves, this is very different from the modern sponge-dominated crinoid communities that you see in Antarctica today. So to conclude, looking through time, you can see that we have varying ecology from informal bivalve deposit communities with predators over the KPG boundary and up here in the Miocene, but then more suspension feeding epifaunal dominated communities at the present day and in the Eocene and the Oligocene. And what we really need to do to further our understanding of what's happening in the evolution of Antarctica is try and better understand the links between the changing climate, the different predator-prey relationships, and what's going on with the depositional environment. Because it may be that the Cape Melville specimens are showing what's happening in deeper waters, whereas all the other things are happening in shallower waters. So there may have been different communities in different environments. So thank you very much.
Okay, thank you, Ron, for your presentation. It was very interesting. And the next one would be by Alexander Kellner, but he's not present here, right? So I pass the word to Fabiana, and she will close the session. Please, Fabi.
Yes, thank you. Just before closing, we want to thank all the authors who accepted to present their work in this session, and we will have a very interesting session next week for discussing all your works and also the works that have not been presented during the web session. Then we would thank all the SCAR organization that allowed us to set up this session, and also in this situation to set up the web session, supporting us in all the steps of its realization. And then we would also to thank our co-conveners, Ron Whittle, who is here, and then Christine Trevisan and Maria Kenya Raffi, that helped us in the revision of all the abstract that we received. And last but not least, I would to thank Fernanda for a helpful and very enthusiastic participation in this initiative. Thank you to everybody.
Thank you very much.
Thank you all, and thank you, Fabiana. I think we did a good job. We were a good team of work. Thank you all.
Bye.
Bye bye.
Bye,
See you
thank
next
you.
week.
Bye.
Bye.